In this class, we are going to study about transcription, gene versus cestron concept, as well as the central dogma of biology. And apart from that, we'll also study about the genetic code. So we'll start from the gene versus cestron concept. In the previous class, we had studied about the replication of DNA, and now we'll study about the expression of genes through transcription and thereafter translation. So before starting transcription, we have to study what is the difference between transcription and replication, and also what is gene and what is cestron. So these two terms are very similar, but they have various different significances in eukaryotes as well as prokaryotes, and also they were postulated at different times. So this gene we have studied in the previous chapter, and we know that gene is a elementary unit of inheritance. It is a passenger of chromosome or passenger of DNA. It is also a functional segment of DNA, and other things about genes we have studied that they have different forms, and those are called alleles. But this cestron concept is new to us, and it has come in this chapter. So first of all, this gene concept was coined by a scientist named Johansson, and he was the first to describe it, and he said that it can be used to replace the Mendelian term factor. In the previous chapter, we had studied about Mendelian factors. So the word factor for Mendel is the word gene for us. So it was done by the scientist named Johansson. So according to him, genes were the units of inheritance, and those were functional segments also. So according to him, the gene was elementary unit of inheritance. But we also saw that Morgan's experiment provided a lot of evidence about what was genes. So according to Morgan's experiment, we have understood that gene is a functional segment of DNA, and also genes could undergo recombination, that is, genes were unit of recombination. So these were the two scientists who first described the gene, but later on two other scientists called Beadle and Tatum proposed something which they called one gene, one enzyme hypothesis. So it was given by Beadle and Tatum. He experimented on something called neurospora. So Neurospora has something called auxotrophs, and those were the nutritional mutants. They were artificially created. And also Neurospora has a natural variety which is called prototroph. So that is not a nutritional mutant. So they experimented on auxotrophs of Neurospora. So accordingly they gave a certain postulate. So they were the first people who postulated that genes control the expression of a character through the formation of an enzyme. We saw in the previous chapter that Mendel was first hinted it, but Mendel had no idea what caused the expression of his Mendelian results. So Mendel was not able to prove how the factors which he postulated affected the traits or characters he studied on pea plant. So for the first time in history of biology, so Beadle and Tatum were the people who first postulated these things, that gene control the expression of a character through the formation of an enzyme. But this was not 100% true. We will see later how. There was another scientist, his name was Janovsky. So in 1967 this happened, and this scientist postulated that one gene could specify only the formation of one polypeptide. And from 11th chapter, that is biomolecules, you must have seen that proteins are made up of polypeptides, and polypeptides are made up of amino acids. So one protein may not be just one polypeptide chain. So there are certain cases where one protein has a lot of polypeptide chains. For example, our hemoglobin, that is our respiratory pigment, that is composed of 4 polypeptide chains, 2 alpha and 2 beta chains. So this scientist Janovsky said that one gene could specify the formation of only one polypeptide, and later on something else happened and that proved that this is actually correct. So we have to understand what is a cestron and what is cestronic concept. So this is what a cestron is. So just like a gene, cestron is also a segment of DNA that contains information. Therefore, cestron is also a functional segment. But apart from that, it has certain differences. That is, it specifies information for formation of a polypeptide tRNA or rRNA. There are also certain features of a cestron, that is, a cestron could also specify formation of certain chemicals, or it could also play a very important role in relation to transcription. So those are the things that differ a cestron from a gene. So nowadays gene is a very old concept. So nowadays more often than not cestron is used in place of gene. So based on the cestronic nature of the DNA, we will see later that eukaryotic DNA is monocestronic and prokaryotic is polycestronic. So now from now on cestron is a more accurate term as compared to a gene. So nowadays cestron is more often used, and we will see later in this chapter that prokaryotic transcription unit is polycestronic and eukaryote, that is ours type, is monocestronic. So these are the normal gene and cestron concept. So we could also infer one specific information from here, is that a gene could have, a normal structural gene could have more than one cestron, that is a structural gene can have more than one cestron. Therefore a gene could cause formation of more than one polypeptide, because one cestron is has resulted in formation of one polypeptide. Therefore if any structural gene contains more than one cestron, that structural gene would have the possibility to create more than one polypeptide chain. And we will see later in the case of lac operon that we have to study in this chapter, that lac operon has polycestronic DNA. Now we come to the main part, that is transcription. But before that we have to understand something, that is the central dogma. So this central dogma states that information flows from DNA to RNA to protein. That is one-way flow of information. So this is the normal central dogma that we know. But also there was another scientist named Commoner. He said that DNA information flows from in a circular way. So that is not in our syllabus, and other is in our syllabus. That is called reverse transcription, and that happens in the case of viruses. So in that case we will study about viruses in the 11th chapter, and there we have to study how and why viruses have a certain different processes as compared to this one. So this is in our syllabus, and this is what we have to study. This process will be called as transcription, and this process will be called as translation. So since it is a crash course class, I am not going into the other details, that is the circular flow of information and the reverse flow of information in this chapter. So we will now study transcription, that is DNA to RNA. Simply speaking, it's transfer of DNA information into RNA. So in transcription, first of all we have to understand what is a structural gene and how it functions. So this could be a long strand of DNA, and there are certain substances or certain molecules attached to it, and those are called promoter or terminator. So a promoter and terminator could be any. This could also be a promoter, this could also be a promoter. But how is that defined? First we have to study that. So if we say this is 5 prime and this is 3 prime and this is 3 prime and this is 5 prime. So there are two strands of DNA over here. So we have to first label which one is a template or which one is a coding strand. So we could label any of them. So if we label this one as a template strand, then we'll first see what happens, and this one as coding strand. So if something has to happen, that is transcription, that is DNA from DNA to RNA, so what we have to do, that a DNA will have to act as a template, and therefore a new RNA strand will be formed in the opposite direction to it. So what happens here is that if this strand is being used as a template strand, whether I have written coding or template, we'll see that later. So if this strand is being used as a template strand, then the new RNA that will be formed will be like this, that is in the 5 prime to 3 prime direction. 5 prime to 3 prime direction. So in that case, since the RNA polymerases will also work in the same direction as DNA polymerase. So we have seen that in the previous class, that DNA polymerase polymerizes only in 5 prime to 3 prime direction, and so is the case with RNA polymerase. So in the case of transcription, first of all we have to know that transcription occurs only due to RNA polymerase. So because of that, RNA polymerase will work in 5 prime to 3 prime direction, and here the complementarity has to be maintained. Therefore what will happen is that this strand will have to act as a template strand, not the upper one. But there is some other case. If we switch the position of promoter and terminator, then the definition of template and coding strand will be just reversed. So therefore any could be template, any could be coding. So in that case, in this case, we have this one as the coding and this one is the template strand. So we'll have to reverse it. And here this will be called promoter, and this is the terminator. So this promoter and terminator, if their positions are switched, then this will become the template and this will become the coding strand. So this template strand is also called the master strand, or it's also called antisense strand. And this coding strand, which does not code for anything, that is called coding strand. So all the nomenclature that is being done over here is with respect to coding strand. So with respect to coding strand, promoter will be at 5' side and terminator will be at 3' side. And now we have to understand what is promoter and what is terminator. So this promoter has 80 rich region called Tata box. It is also called Prinbo box. Prinbo is the name of the scientist who discovered it. So it has adenine thymine rich region, and that is why it becomes depressed, and we'll later see what and how that happens. This terminator part has polyadenyl region. It has poly A region. So this is a basic transcription unit. It contains a promoter, a terminator, and from promoter to terminator, the main part is called something which is called a structural gene. So this part, from a promoter to a terminator, it is called structural gene. So this promoter, structural gene and a terminator together constitute the transcription unit, and this promoter has a 80 rich region and terminator has poly A region, that is polyadenosine region. So we'll study here transcription unit. So a transcription unit has a promoter plus a structural gene plus a terminator. But in eukaryotes, apart from the promoter, there is also something called enhancer. But since enhancer is not in our NCERT syllabus, I am not teaching you that here. So that promoter and enhancer would also be here in the case of eukaryotes. But it's general, the normal transcription unit consists of a promoter. All these things are labeled according to the coding strand, that is with respect to the coding strand. So this promoter is on the 5' end of the coding strand and terminator is on the 3' end of the coding strand. So although its name is coding strand, but it does not code for anything because only one or this template strand is being transcripted. So unlike replication, here only one strand of DNA will be transcripted, not like replication that both the two strands will be replicated. So in replication we had seen that both the strands were replicated, and also when replication begins at one part, it proceeds to the whole DNA's part. But here what is happening is that only one strand, either this or this, so that could be reversed also. So either of the two strands will be transcribed. That is the first point. And the second point over here is that only a segment, not the whole length of one single strand, whether it be the coding or the template strand, just one small segment would be transcribed in the 5' direction to the 3' direction with respect to the coding strand, not with the in respect to the template strand. So these are the basic differences in between transcription and in between replication. So in replication the whole DNA was replicated, here only a segment will be replicated. In replication both the strands, here only one strand. So this is the basics of transcription. Now we will start the main process of transcription. So the first step in transcription is the activation of nucleotide. We just saw in the replication that first of all the nucleotides were transformed from monophosphate to the triphosphate. So the first step would be activation of substrates. So what happens here is that adenosine monophosphate would be transformed into adenosine triphosphate. We had seen in the case of the DNA replication, what happens is that deoxyadenosine monophosphate was used into deoxyadenosine triphosphate, but here DNA is being transformed into RNA, so the substrate would be not deoxy but normal AMP, and that will be transformed into ATP. Likewise with CMP, CTP, GMP, GTP and TMP, TTP. So the monophosphates are transformed into triphosphate. And we also know from our chapter of respiration that triphosphates are a source of energy. So all these are used as substrates and also as a source of energy for the transcription process. So this is the first step. Now in DNA replication we had seen that the DNA strand has to be opened, but here the whole DNA strand does not have to open, only a small part will be opened. So this is the second step in DNA replication. We had also seen that helicase or unwindase was the one which was opening the DNA strand. Now what will happen is interesting. Now RNA polymerase would bind in the region of the promoter, and thereafter something will be initiated. So this RNA polymerase, that is the main transcripting enzyme, this will associate with something called sigma factor. And what is this sigma factor? This is the initiation factor for transcription. Without sigma factor, RNA polymerase cannot bind in the promoter region. We have just seen what is promoter region. So in that region, the RNA polymerase would associate with sigma factor and initiate transcription. So what happens here is that the chain elongation property of RNA polymerase is the only property that RNA polymerase has. So RNA polymerase cannot initiate in a transcription. RNA polymerase cannot initiate termination. That is, it neither has the ability to terminate or initiate transcription. So its association with the factor sigma and the rho decides it. So how does it do chain elongation? So in this case, what happens is that RNA polymerase, if this is the DNA strand, we will study how this occurs. So if it is the DNA strand and it has certain regions, so the new strand of RNA that will be formed would be on the inner side of, and this is the opened part of the DNA. Initial part of the DNA or the later part of the DNA would be coiled as usual. So if it is here A, then the new RNA molecule which is being formed here, if this is the template one, then that will have U over there. If it has G, it will have C. If it has C, it will have G. So normally in DNA replication, A is in adjacent to T, but here U is in opposite to A, because in RNA U is replaced by T, that is uracil is replaced by thymine. So this is the normal form that occurs, and its diagram is also given in NCERT. You can very well see it from there how this occurs. So RNA polymerase can only cause the chain elongation. Now we come to the last part. So RNA polymerase first associate with sigma, then alone, and then with the rho factor, and it is also the termination factor, and this will cause in the termination, that is stoppage of transcription. So this process occurs in differently in bacteria, that is prokaryotes, and the other eukaryotes. What happens here is that prokaryotes have only a single RNA polymerase that catalyzes the formation of all types of RNAs. So in prokaryotes, So we will see later that there are three types of RNA, that is mRNA, rRNA and tRNA. So in prokaryotes, a single RNA polymerase enzyme is capable of synthesizing all those types of RNAs. But in eukaryotes it is very different. In eukaryotes, there are three different types of RNA polymerases. First is RNA polymerase 1, the other is 2, so these are different types of RNA polymerase, and these are responsible for formation of different types of RNA. So the polymerase 1 enzyme forms rRNA, that is, it has various different sized subunits, and those are 28S, 18S and 5.8S. This RNA polymerase forms something called hnRNA, that is called heterogeneous nuclear RNA, and that is transformed, that will later form something called mRNA. This third part forms something called tRNA, and also it forms a subunit of 5S rRNA. So these are the three types of enzymes, and these each form different type of RNA. So this is 5S rRNA, and that is a type of this rRNA. So normally only RNA polymerase 1 forms the RNA, but this is an exception. So these are the smaller parts. So this rRNA is initially formed in a large subunit that is breaked or that causes a cleavage, and therefore these subunits are formed. So this is the normal layout of transcription in eukaryotes. So we also have to study these various types of RNAs and what are their properties. So the primary transcript of RNA, that is the initial RNA or raw RNA that is formed, that has to be modified to form functional RNA. So we will now study about post-transcriptional modification. So first of all, we will have to study cleavage. So what happens here is that large sized rRNAs are broken down. So what happens here is that the initial subunit that is formed of 40S or 41S, so that will be broken to form two different sized rRNAs, and one is 32 and the other is 20S or 20S. So this will also be broken down to form 28S as well as 5.8S. This will also be broken down to form something called 18S. S is the sedimentation coefficient or the Schwarzschild coefficient. So we have to study that in 11th class what is S, that is the sedimentation coefficient. So this is an indirect measure of the size of the ribosome. So this is about rRNA. We also have to study about mRNA, what happens with the case of mRNA. So we first start with splicing, and we also have to study about capping and tailing. So first we will study about capping and then tailing. So in mRNA what happens is that a lot of other things apart from the main required things are present. So in DNA there are certain sequences called introns and exons, and capping and tailing are the post-transcriptional modifications, and also something called splicing occurs because of those introns and exons. So in DNA there are something required and unrequired segments also. So what happens here is that this black portion is something called exons, and this red portion is something called introns. So these portions are the functional portions. Functional portions. So if RNA is transcribed from it, that is, if after transcription it forms mRNA, so if it forms mRNA, that will also have these things, that is introns and exons. So we also have to label the polarity, that is, this is the 5' end and this is the 3' end. So this is DNA and this is HN mRNA. Because it is not mature mRNA, we will call it HN RNA, that is heterogeneous nucleus RNA. So in this case, first of all, this will also have introns and exons. So these are the intron portions, that is the red one, and here also there are introns. So that has to be cut, but before that something else happens, and that is called capping. So in capping what happens is that an unusual base, that is 7-methyl guanosine, is added over here. So this is 7-methyl guanosine. Apart from that, another process has to occur over here, and that is called tailing. So what happens in the case of tailing is that this mRNA will have a long tail. So this tailing process that occurs has to happen on the 3' end, and this capping process occurs on the 5' end. So in this case a polyadenyl tail is added. So this was the 3' end and this was the 5' end. Here a methyl guanosine group is already present. So this is the 7-Mg group. So after these processes something else has to happen, and that is called splicing. So splicing is removal of these introns and joining of those exons. These are non-functional segments and these exons are functional segments. So for that something else has to happen. So in this region something attaches and which is called snRNP. Those are called small nucleoriboproteins. So this is SN, small nuclear ribonucleoprotein, or these are called SNRPs. So this will cause this splicing, and therefore now the mRNA will become smaller and only it will have black segments. So this is mature mRNA. So this process is known as splicing, and it occurs with the help of spliceosomes. So this process from year to year is called splicing. So this is the processing of mRNA, that is hnRNA from mRNA. The first process that occurred was capping, then tailing, then splicing, and therefore it results in the formation of mature mRNA or the messenger RNA. This is transported out of the nucleus into the cytoplasm. So we'll study about here that so capping is addition of 7-methyl guanosine at 5' end of hnRNA. The third part is tailing. It is addition of 200 to 300 poly A tail. So we just have seen what is poly A tail, and that occurs at the 3' direction. After that, splicing. It involves removal of introns and joining of exons. So this is what happens over here. This intron has been removed by SNRPs. So an intron is a non-functional segment and exons are functional segments. So is it that introns were uselessly present from the beginning? No, it is not like that. Introns were functional in our ancestors, and therefore the presence of introns is then reminiscent of antiquity. That is, in our ancestors, these introns were functional segments. But now in ours, that is in our generation or in our species, that has become non-functional. So it is a feature of our ancient genome, and this splicing that is occurring over here, this shows the dominance of RNA world in our ancestors. So this splicing process and this introns are a very strong evidence that RNA precedes DNA. These provide very important evidences that RNA came much before much before DNA. So these are the normal points about this. So now we will start various types of RNAs that we have to study, or the main types of the RNAs are rRNA, mRNA and tRNA. Apart from that, we also have to study certain other types of RNAs like genomic, catalytic or cytoplasmic RNA, but normally we have to limit our discussion to the ribosomal RNA, mRNA and the tRNA. So the first type of RNA that we have to study is the rRNA. It is called R because it is a constituent of ribosomal RNA. So it is found in ribosomes. So it is found attached to proteins inside ribosomes. Apart from that, we also have to study about that various components or the various small parts of rRNA. So in prokaryotes, there are various parts of RNA, so that are 5S rRNA. Apart from that, there is 16S rRNA and also 23S rRNA. So in eukaryotes, there are various subunits also. In eukaryote there is also 5s rRNA. Then we also have 5.8s rRNA. Apart from that, we also have 18s rRNA and also 20s rRNA. So this is the difference between prokaryotic and eukaryotic rRNA. So generally ribosomes have two subunits, that is one larger subunit and the other smaller subunit. So the larger subunit in prokaryotes, ribosomes together constitute 70s ribosomes. In prokaryotes, ribosomes are 70s ribosomes. So these are divided into larger subunit and smaller subunit. So this larger subunit contains this one 5s RNA and 23s rRNA. In smaller subunit, 16s rRNA is present. Now in eukaryotes, the ribosomes are 80s. So here also there is two subunit. The first is larger. It is 60s. And here in the case of bacteria, in the case of prokaryotes, the larger subunit is of 50s and the smaller subunit is of 30s. In the case of larger subunits of prokaryotes, eukaryotes, we have 80s ribosomes and the smaller subunit consists of 40s. So the larger subunit, that is 60s out of the 80s ribosomes in eukaryotes, contains 5s rRNA, 5.8s rRNA, as well as 28s rRNA. But the smaller subunit contains this 18s rRNA only. So ribosomal RNAs are present inside the ribosomes and these are attached to the ribosomal proteins. So this was the normal thing about ribosomal RNA. We will also study the differences between rRNA, tRNA and mRNA later. Now the other RNA, that is the tRNA. It is also called transfer RNA or soluble RNA. So we have to study two models of it. First one is 2D model. (2D model is also called clover leaf model because it looks like a clover leaf model. So this is a normal 2D clover leaf model of tRNA. We'll also have to label the parts. So this part or this side is called T-Cy-C loop. So this part is called T-Cy-C arm. This is called anticodon arm. And this is called DHU arm. So this Cy word stands for pseudouridine and this DHU stands for dihydrouridine. So we don't have to study the details of these DHU and T-Cy-C, but we have to remember what are those there and functions. And the upper side, that is at the 3' end, it is the amino acid acceptor side. Amino acid acceptor arm. So these are the 4 sides, and now we have to understand what these contain. So this anticodon arm contains, these are the 3 nitrogenous bases that are present, and these have the information for a certain specific amino acid. So this anticodon loop has bases that codons, it contains codons, and these are complementary to codons on mRNA. So we will later study what is this codon, because we will study that in genetic code. And this T-Cy-C arm is the arm through which it attaches to ribosome. And this DHU arm that is here, so these are the 4 sites and they have their own specific functions. This thing, the small projection over here, is a vestigial part, or it is a non-functional part, and scientists do not know about the function of this part. So this is called a rudimentary form, or you could also say it as variable arm. This is the anticodon loop. This is the amino acid acceptors arm. This is the DHU arm. So these 4 have their different functions, but apart from that we also have to study the 2D model or the 3D model. So the normal inverted L type of diagram is already given in the NCERT, and that is really tough to draw with hand, and therefore I am not drawing it over here. But we have to study that this in 3D state tRNA looks like an inverted L, and there also the arms would be same, but their orientation will look different in the case of 3D model. than in the case of 2D molecule. So this is about tRNA, and tRNA is specific for each amino acid. So we have to remember this. So when translation occurs, each tRNA picks up its own amino acids, and therefore it carries that amino acid to the ribosomes for their attachment. There are more than 100 types of RNA, tRNA. So there is a separate type of tRNA for the initiator codon, that is the AUG, and but no specific initiator codons or any sort of termination codon occur for the termination codon, that is AUG, GUG and other parts. So we will later see that. So these are the basic information about tRNA. So tRNA is now called transfer RNA, but before the discovery of something called genetic code, it was called soluble RNA or soluble RNA in general. So before, initial name is soluble RNA, modern name is transfer RNA. After the discovery of genetic code, its name was changed from soluble RNA to transfer RNA. Now we have to see the mRNA. The third type is mRNA. It's also called messenger RNA. It's also called informational RNA. Or it's also called genetic RNA. So we'll first study its structure. We had seen a few moments ago. So this is the main coding part, and the first and the termination codon are there. So this red shaded portion is the codon part. Codon is something which we will understand after we study the genetic code. So this is the initiator codon. This is the termination codon. This is the 7 mg cap, and this is the poly A tail. So these are the normal parts of a DNA. So this green shaded portion is the main portion which we called as coding region. So this is the main informational part of mRNA. This is the non-coding leader region. This is the 5 prime end, and this is the 3 prime end, and this is also a non-coding region which is called trailer region. This is the 3 prime end of it, and the green colored part is the main coding region. Now the first codon that occurs is called the initiator codon. So this is leader region, and this initial part and this ending part is the initiator and termination codon. And the first part after the cap is the non-coding leader region. So these are the normal parts of mRNA. It is also called informational or messenger RNA because it carries the message, the main sequence of codons which code for the sequence of polypeptide or the amino acids of a polypeptide. So it acts as a template during translation. That is, when translation or protein formation starts, it acts as a template, just like a strand of DNA. So these were the 3 main types of RNAs that is in our syllabus. So we also have to study their differences. Now we will study their differences. So the first criteria is size, then we have stability, then we have abundance, so on the basis of these we have to understand the relative nature of these 3. So mRNA is the heaviest or the largest in terms of size. rRNA is also very large, but it's smaller than this normally. It is the smallest RNA, that is the tRNA. If we talk in terms of stability, it is least stable. It is the most stable, and it lies in somewhere in the intermediate range. That is between most and least stable tRNA occurs. If we talk about the abundance, it is the least abundant, only 3 to 5% in an active cell. This rRNA is the most abundant, approximately 80%, and this is intermediate. In terms of abundance, it's around 15%. So these are the three main differences between the mRNA, rRNA and tRNA: size, stability and abundance. After that, we also have to study about a few small points like some other types of RNA like genomic RNA. So apart from that, we also have to study a few other types of RNA, and we'll see that in brief. So the fourth type of RNA is scRNA. It's called small cytoplasmic RNA. It attaches ribosomes to endoplasmic reticulum. We also have something called catalytic RNA. So these RNAs function as enzymes. Example is peptidyl transferase, etc. We also had seen a RNA enzyme which we said as ribonuclease when we were studying cleavage. After that, we also have to study something called genomic RNA. Here RNA acts as the genetic material, that is when we studied the difference between RNA and DNA, we had studied that RNA acts as the genetic material in some viruses. So this is that genomic RNA. RNA acts as genetic material. Example is riboviruses. So these were some other types of RNAs, but we mainly have to study the mRNA, tRNA and rRNA. So now we have completed the types of RNA and the transcription. So after that, we'll start the genetic code and thereafter translation.